Hello everyone, welcome back to the channel. In the previous video, we discussed about the vector-mediated gene transfer to plants. In this video we will learn about the vectorless mode of genetic transfer in plants. And also about the edible vaccines. Direct or vectorless DNA transfer. The term direct or vector-less transfer of DNA is used when the foreign DNA is directly introduced into the plant genome. Direct DNA transfer methods rely on the delivery of naked DNA into the plant cells. This is in contrast to the agrobacterium or vector-mediated DNA transfer, which may be regarded as indirect methods. Majority of the direct DNA transfer methods are simple and effective. And in fact, several transgenic plants have been developed by this approach. Types of direct DNA transfer. The direct DNA transfer can be broadly divided into three categories. 1. Physical gene transfer methods, electroporation, particle bombardment, microinjection, liposome fusion, silicon carbide fibers. 2. Chemical gene transfer methods, polyethylene glycol mediated, diethyl amino ethyl dextran mediated, calcium phosphate precipitate. Physical gene transfer methods. Electroporation. Electroporation basically involves the use of high field strength electrical impulses to reversibly permeabilize the cell membranes for the uptake of DNA. This technique can be used for the delivery of DNA into intact plant cells and protoplasts. In this method, the plant material is incubated in a buffer solution containing the desired foreign target DNA and subjected to high voltage electrical impulses. This electrical pulses causes the formation of pores in the plasma membrane through which DNA enters and gets integrated into the host cell genome. In the early years, only protoplasts were used for gene transfer by electroporation. Nowadays, intact cells, callus cultures, and immature embryos can be used with suitable pre- and post-electroporation treatments. Electroporation has been successfully used for the production of transgenic plants of many cereals, e.g. rice, wheat, maize. Advantages of electroporation. This technique is simple, convenient and rapid, besides being cost-effective. And also, the transformed cells are at the same physiological state after electroporation. Efficiency of transformation can be improved by optimizing the electrical field strength and addition of spermidine. But this method also has limitations. Under normal conditions, the amount of DNA delivered into plant cells is very low. Efficiency of electroporation is highly variable depending on the plant material and the treatment conditions. Regeneration of plants is not very easy, particularly when protoplasts are used. Another method of physical mode of genetic transfer is particle bombardment, which is also called as biolistics. Particle or microprojectile bombardment is the most effective method for gene transfer and creation of transgenic plants. This method is versatile due to the fact that it can be successfully used for the DNA transfer in mammalian cells and microorganisms. The microprojectile bombardment method was initially named as biolistics by its inventor Sanford, 1988. Biolistics is a combination of biological and ballistics. This method is called by several names such as particle gun, gene gun, bioblaster. Microcarriers or microprojectiles, the tungsten or gold particles coated with DNA, and those are carried by macrocarriers, macroprojectiles. These macrocarriers are inserted into the apparatus and pushed downward by rupturing the disc. When it pushes downward, the stopping plate does not permit the movement of macrocarrier while the microcarriers, with DNA, are propelled at a high speed into the plant material placed below in it. Here the DNA segments are released which enter the plant cells and integrate with the genome. In order to protect plant tissues from being damaged by bombardment, cultures are maintained on high osmotic media or subjected to limited plasmolysis. Advantages of particle bombardment. Gene transfer can be efficiently done in organized tissues. Different species of plants can be used to develop transgenic plants. Then what are the limitations of particle bombardment? The major complication is the production of high transgene copy number. This may result in instability of transgene expression due to gene silencing. The target tissue may often get damaged due to lack of control of bombardment velocity. Sometimes, chances of regeneration of undesirable chimeric plants is also there. Let's move to the next method. Microinjection. Microinjection is a direct physical method involving the mechanical insertion of the desirable DNA into a target cell. The target cell may be the one identified from intact cells, protoplasts, callus, embryos, meristems, etc. Microinjection is used for the transfer of cellular organelles and for the manipulation of chromosomes. The technique of microinjection involves the transfer of the gene through a micropipette of 0.5 to 10.0 picometers tip into the cytoplasm nucleus of a plant cell or protoplast. While the gene transfer is done, the recipient cells are kept immobilized in agarose embedding and held by a suction-holding pipette. As the process of microinjection is complete, the transformed cell is cultured and grown to develop into a transgenic plant. In fact, transgenic tobacco and Brassica napus have been developed by this approach. The method is looking quite easy, but it is slow, expensive, and has to be performed by trained and skilled personnel. 4. Liposome-mediated transformation. Liposomes are artificially created lipid vesicles containing a phospholipid membrane. They are successfully used in mammalian cells for the delivery of proteins, drugs, etc. Liposomes carrying genes can be employed to fuse with protoplasts and transfer the genes. The efficiency of transformation increases when the process is carried out in conjunction with polyethylene glycol. Liposome-mediated transformation involves adhesion of liposomes to the protoplast surface, its fusion at the site of attachment and release of plasmids inside the cell. This method is mainly dependent on the principle that protoplast contains lips membrane, since its lack in cell wall. Lipid vesicles also has lipid, helps in rapid adhesion to protoplast membrane and helps in gene delivery. Advantages of liposome fusion are: being present in an encapsulated form of liposomes, DNA is protected from environmental insults and damage. DNA is stable and can be stored for some time in liposomes prior to transfer. Applicable to a wide range of plant cells. There is good reproducibility in the technique. But this method also has some limitations. The major problem with liposome-mediated transformation is the difficulty associated with the regeneration of plants from transformed protoplasts. 5. Silicon carbide fiber-mediated transformation. The silicon carbide fibers, SCF, are about 0.3 to 0.6 picometer in diameter and 10 to 100 picometer in length. These fibers are capable of penetrating the cell wall and plasma membrane and thus can deliver DNA into the cells. The DNA-coated silicon carbide fibers are vortexed with plant material, suspension culture, calluses. During the mixing, DNA adhering to the fibers enters the cells and gets stably integrated with the host genome. The silicon carbide fibers, with the trade name whiskers, are available in the market. Advantages of SCF-mediated transformation. Since the DNA delivery is directly into intact walled cells, This avoids the protoplast isolation. Procedure is simple and does not involve costly equipment. Chemical gene transfer methods. In chemical employed gene transfer, the major one is polyethylene glycol mediated transfer. Polyethylene glycol, PEG, in the presence of divalent cations, using Ca2+, destabilizes the plasma membrane of protoplasts and renders it permeable to naked DNA. In this way, the DNA enters nucleus of the protoplasts and gets integrated with the genome. The procedure involves the isolation of protoplasts and their suspension, addition of plasmid DNA, followed by a slow addition of 40% PEG 4000 Wv, which is dissolved in mannitol and calcium nitrate solution. As this mixture is incubated, protoplasts get transformed. The advantages of PEG-mediated transformation are: a large number of protoplasts can be simultaneously transformed. This technique can be successfully used for a wide range of plant species. Limitations of PEG-mediated transformation are: the DNA is susceptible for degradation and rearrangement. Random integration of foreign DNA into genome may result in undesirable traits. Regeneration of plants from transformed protoplasts is a difficult task. Diethyl amino ethyl dextran mediated transfer. In this method, desirable DNA can be complexed with a high molecular weight polymer diethyl amino ethyl dextran and transferred. The major limitation of this approach is that it does not yield stable transformants. Calcium phosphate co-precipitation mediated transfer. The DNA is allowed to mix with calcium chloride solution and isotonic phosphate buffer to form DNA calcium phosphate precipitate. When the actively dividing cells in culture are exposed to this precipitate for several hours, the cells get transformed. The success of this method is dependent on the high concentration of DNA and the protection of the complex precipitate. Addition of dimethyl sulfoxide, DMSO, increases the efficiency of transformation. One of the most advanced and useful application of the transgenesis. Edible vaccines. Edible vaccines are nothing but transgenic plant and animal-based production of or those that contain agents that trigger an animal's immune response. The first successful human trial of an edible vaccine was conducted in year 1997, in which volunteers were fed transgenic potatoes. This potato contains the B subunit of the E. coli heat labile toxin, responsible for diarrhea. When they ate this, they will develop immunity against diarrhea. How these edible vaccines are produced. First, the gene responsible for particular disease or responsible for immune response, which simply called as antigenic genes, were placed into the plant cell through any one of the method discussed earlier. When the plants grown, they also grown with the inserted antigenic genes. When we consume that plant which has antigenic genes, body will treat that gene as antigen and develops immunity against it. Advantages of edible vaccines are: they are cheap, therefore they can be mass-produced. As any other commercial antigens needs to be isolated and purified, but in this case, they can be ingested by eating the plant part of the pool plant. So, the need to process and purify does not arise. Extensive storage facilities like cold storage are not required for storage like of other drugs. If the local native crop of a particular area is engineered to produce the vaccine, then the need for transportation and distribution can be eliminated. Most importantly, they trigger the immunity at the mucosal surfaces such as those that line the mouth, mucosal immunity, which is the body's first line of defense.